Hi everyone, it's a great pleasure to have this opportunity to share some of our work on VGB. So the title of this talk is VGB, the yin and yang in angiogenesis, and my name is Xuri. At least in China, we believe that all things exist as inseparable and yet contradictory opposites. For one example, the females and males each has some qualities of the other side, and this way everything's balanced very well. And based on our work and others' work as well, and we think that VGB has such qualities. Depending on the specific conditions, it can be pro-angiogenic in blood vessel during blood vessel death. On the other hand, it can also be anti-angiogenic during blood vessel growth. So in this talk, I'm going to explain to you why we think this is the case. So what VGB is? It's a growth factor discovered in 1996. It has a very high homology to VEGF-A, which is better known in the field. VGB binds to VEGF receptor 1 and neuropilin 1. As shown here, you can see that the receptor binding domain of VGB has almost 50% homology with VEGF-A. As such, upon its discovery, VGB was initially believed to be an angiogenic factor for a very long time. However, along that line, all our work trying to find out the angiogenic effect of VGB were all negative. This is to show you what we did. For example, in VGB knockout mice and transgenic mice, in the blood vessel growth, cell migration assays, and permeability, and all these animal models, we didn't find any angiogenic effect of VGB, and we were very, very disappointed. So, for a very, very long time, we were really puzzled. What is the function of VGB, and does it really do anything at all? It's until 2000, we did the myocardial infarction animal model, and only under this pathological condition, and we found that VGB is needed for the survival of blood vessels. You can see that when you give VGB by adenovirus expression, you can see more surviving blood vessels. And subsequently, we looked at the mechanism, how VGB has this pro-survival effect. By RNA sequencing analysis, we found that VGB inhibits the expression of many, many apoptotic factors. It inhibits the expression of these apoptotic factors by nearly tenfold, as you can see here. So, does VGB really promote VGB blood vessel survival or not? So, to answer that question, we use a cornea pocket assay. Now, I need you to understand that in this cornea pocket assay, there are two different phases. It's not really simply just one animal model, because the two phases tell different things. The first phase is that when you give growth factors, in this case FGF2, the growth factor can induce the growth of blood vessels from the pre-existing one, like this one. These are new blood vessels. So this is the first phase, the blood vessel growth phase. However, if you take away the growth factor, you implant it, These blood vessels, they won't stay, they disappear over time. And this is the second phase, the blood vessel survival phase. There's nothing growing, okay? What you are seeing is how the blood vessel degenerate or survive. So, in the second phase, using this model, we can see that without VGB, this is VGB knockout mice, these blood vessels, they don't survive well, and without VGB, you can see less blood vessels. However, it is important to remember that this is the survival phase, not the growth phase. So, VGB is critical for blood vessel survival. Now, how about the role of VGB in pathological angiogenesis? So to address that question, we used a laser-induced choroidal neovascularization model. In this model, this is a well-established model and it is published by many experts. Now, this article shows that in this model, between day 0 and day 5 after laser injury, there is a growth of new blood vessels. However, after day 5, you can see that the new vessels, they disappear gradually over time. So if you look at blood vessels before day 5, you're looking at blood vessel growth. If you look at blood vessels during day 5 and day 28, you actually are not looking at growth of blood vessels, but survival of blood vessels. So using this model at day 7, we found that if we inhibit VGB by shRNA or by neutralizing antibody, the choroidal blood vessels are decreased. So inhibit VGB in this case can inhibit CMV. But since this is day 7, as I said, this probably is because of the reduced survival of blood vessels instead of growth. And subsequently, we used another disease model, the retinitis pigmentosa disease model, which is RD1 mice. It is well known that in these mice, the retina start to degenerate after postnatal day 11, I believe. So in this model, if you give VGB here, you can see that you can have more blood vessels, the green ones, and without VGB, very few blood vessels left due to the retinal degeneration. This is HE staining showing that if you give the mice VGB, the retina, to a large extent, is rescued from degeneration, while the BSA group, you see the retina is severely degenerated and then there is hardly any outer nuclear layer. So, VGB can rescue blood vessels from degeneration, at least in this RD1 mice model. Now, the first part of my conclusion is that VGB can be a pro-angiogenic and pro-survival factor during tissue degeneration, because when everything's dying, there's no other growth factors, and that's why they degenerate. And this pro-angiogenic and pro-survival effect of VGB, because it can promote blood vessel survival and inhibit apoptosis and oxidative stress. And that is the first aspect of VGB I want to talk about here. So during blood vessel degeneration, VGB can be pro-angiogenic, the yin aspect of VGB. However, those research doesn't really answer all the questions. There are still many confusing observations exist. For one example, this is Swedish group. They published a paper showing that if you overexpress VGB here, it actually suppresses tumor growth by 50%. If you don't have VGB here in this group, and the tumors grow like crazy, it's twice as big as the control group. So, as a growth factor, VGB actually is inhibiting tumor growth. Isn't this very, very strange? Another growth group also published that in human xenograft tumors, if you overexpress VGB, it inhibits tumor growth. This line. And VGB also inhibits tumor angiogenesis with less blood vessels here. So, this also goes against what I just told you. VGB actually here is inhibiting blood vessel density and tumor growth. So we looked at literature to see how is VGB expressed in cancer patients, and we found that actually there are many publications showing that in cancer patients, low VGB level is associated with very bad survival, and high VGB level is associated with good survival, meaning that VGB may be needed to inhibit tumor growth even in humans. And in this publication, it even shows that in breast cancer patients, the VGB level in their serum is much lower. And this is the analysis using NIH database, also showing that in breast cancer, pancreatic cancer, and esophagus cancer, at least in these patients, if their VGB level is low here, they have very bad survival. Low VGB level, bad survival. Low VGB level, bad survival, also supporting that VGB is needed to inhibit tumor growth. So, how to understand this? As a growth factor, does VGB really inhibit angiogenesis and tumor growth? So, the next half of my talk is about the other aspect of VGB, the young aspect of VGB angiogenesis. So what I'm going to show you that, yes, VGB sometimes really is anti-angiogenic and anti-tumor growth. So here is the evidence, first evidence. Since we want to know how VGB works, we screened a phosphotyrosine kinase antibody array to try to find the unknown mechanisms, and we found that VGB interacts with FGF receptor 1. That came as a huge surprise because we know that everybody knows that VGB binds to VEGF receptor 1 and neuropilin 1. That's it. So this shows that VGB actually can bind to FGF receptor 1, which could be new receptor for VGB. And this is Vicore assay. It shows that VGB really binds to FGF receptor 1 here with very high affinity, almost the same as the classic ligand FGF2, while the other growth factor, PLGF, didn't bind anything at all. So the binding VGB to FGF receptor 1 is specific. This is just another assay, ELISA assay, showing that VGB binds to FGF receptor 1 and compete with FGF2 for FGF receptor 1 binding. So the binding is real and it's very high affinity. So the next question is, which domain of this receptor does VGB bind to? So to answer this question, we made recombinant proteins of different domains of this receptor, and we performed the binding analysis, and we found that VGB mainly binds to FGF receptor 1 domain 2 and 3 instead of domain 1. It's very high affinity, and very low affinity. Since we know that VGB binds to VEGF receptor 1, So does it induce the complex formation of FG receptor 1 with VG receptor 1 or not? First, in mouse tissues, we look at whether this complex exists or not. We found that if we immunoprecipitate FG receptor 1, we can find VG receptor 1 with it, indicating the natural existence of FG receptor 1 and VG receptor 1 complex. Now, encountered endothelial cells expressing both of these receptors, if we give it more VGB, we can see more complex formation. So VGB does induce the complex formation of these two receptors. So we wanted to know whether VGB binds to the heterodimer or not. So we made recombinant heterodimer of VG receptor 1 and FG receptor 1 using the extracellular domain of these two receptors. And this is just to show the recombinant proteins of these heterodimer receptors. And bioassay shows that VGB does bind to the FG receptor 1 and VG receptor 1 heterodimer with a very high affinity, higher than its affinity with FG receptor 1, FG receptor 1 homodimer. No matter the order of the heterodimer is, if it is VG receptor 1 followed by FG receptor 1, or the other way around, FG receptor 1 followed by VG receptor 1, VGB also binds to all of them with very high affinity. So, what's the biological significance of this act of VGB? This is a matrigel assay. It shows that if you give FGF2 and implant the matrigel in mice, you see tons of blood vessels here. If you give VGB together with FGF2 here, now you see very few blood vessels. Shows that VGB actually inhibits FGF2-induced angiogenesis almost completely. How about in mice in vivo? So we next use VGB knockout mice to look at the effect of VGB. Now there are different types. There are 3 types of VGB knockout mice in the world. These 2 are published, the Australian group and the Swedish group. And they knocked out just 1 exon here and a few exons here. And in our knockout, which is made by the Jackson lab, nearly all the exons were deleted. Apart from the knockout mice, we also use the VGB knockout rats from Xuri and Litolo's lab. So using both knockout mice and rats, we look at the blood vessel density. And this shows that in VGB knockout mice, in the retina, the blood vessel density here without VGB actually is higher. More blood vessel branch points and more blood vessel area. So genetic deletion VGB leads to more blood vessels. And this is the VGB knockout rat. Also in retinal blood vessels without VGB here, you can see that the vascular zone is higher, even though the whole retina area is not affected. So again, genetic deletion VGB leads to higher blood vessel density. So what's the molecular mechanism? We screened phospho-MAP kinase antibody array and found that VGB appears to inhibit ERK activation and encountered in the endothelial cells. So this shows that now FGF2 induced ERK activation here, and VGB didn't do anything. And if you give VGB together with FGF2, the FGF2 induced ERK activation is significantly reduced. This is in vivo in mouse retina. Once again, FGF2 activated ERK. VGB didn't do anything. And FGF2, when given VGB, the ERK activation is reduced. And VGB didn't affect the VEGF-A induced ERK activation. So we checked what's the gene regulated by VGB. And microanalysis showed that VGB increased expression of SPRI4 and Western blot validated. If you see VGB, the SPRI4 protein level increases. And it is known that SPRI4 is an inhibitor of the FGF receptor pathway. And what SPRI4 does is that it binds to the signaling molecules and to inhibit FGF2-induced ERK activation, and therefore the function of FGF2. So we next use SPRI4 knockout mice to test whether the inhibitory effect of VGB is really mediated by SPRI4 or not. Now, in SPRI4 wild-type retina, once again, FGF2 increased ERK activation. If you give VGB, the FGF2-induced ERK activation is reduced. However, in SPRI4 knockout mice, now the inhibitory effect of VGB diminished, showing that the inhibitory effect of VGB really requires SPRI4. Now, in summary, our working model is that when FGF2 and FGF receptors are highly expressed, VGB can be anti-angiogenic to inhibit the FGF2, FGF receptor 1 pathway. There are two mechanisms. One mechanism is that VGB can induce the heterodimer formation of FGF receptor 1 and VEGF receptor 1, and to increase SPRI4 expression and therefore inhibit ERK activation. Another mechanism is that VGB can bind directly to the FGF receptor 1 to compete off FGF2 binding, and therefore inhibiting this pathway. So, the second part of my conclusion is that VGB is anti-angiogenic, at least when FGF2 and FGF receptor expression are very high, for example, in tumors. This way, VGB can inhibit the FGF receptor pathway. So, in summary, our work and other work showing that VGB can be both pro-angiogenic and anti-angiogenic depending on the specific context. For example, when blood vessels are degenerating and dying, VGB actually is there to protect them, to survive better, and you can see a pro-angiogenic effect of VGB. On the other hand, when the blood vessels are overgrowing, for example, there are very high level of FGF2 and FGF receptors, and then VGB is there to inhibit the overgrowth of blood vessels so that everything is normal. So this is how VGB works. So our findings showing that the role of VGB is context dependent. Depending on the specific context, sometimes its function can be even opposite. And therefore, it is very important to understand the context and the specific conditions one is looking at. And this really may explain the diverse findings on VGB from different laboratories and using different models over the years. It has been very confusing to many people because different labs have reported different observations and sometimes opposite, as I said. I think our work may explain some of them at least. So, is there any clinical relevance? Yes. VGB right now is being inhibited together with the other VJ family members, of course, in different patients with different neovascular diseases, such as ocular neovascular diseases and different cancers, etc. However, when FGF2 and FGF receptors are highly expressed, inhibition of VGB really should use caution because VGB is there to inhibit the FGF2 and the FGF receptor signaling pathway. Okay, so that's all about my talk today. With that, I would like to thank all the members in my team who have been working really hard, without whom I would not be able to have this opportunity to show all these exciting results. Thank you, and bye.